Biologists usually call ATP the universal currency of energy, but what does that actually mean? To understand, let's dive deeper into the life cycle of ATP from how it is produced to how it is used and finally how it is recycled. So first of all, ATP or adenosine triphosphate is a molecule that provides energy to support processes at cellular level. Structurally, it is composed of a nitrogenous base, a ribose sugar, and three phosphate groups. But how is ATP created? The process of ATP production begins with food. There are many varieties of food. Some of it are carbohydrates, some of it are fats, and some of it are proteins. And when we eat, these macromolecules are broken down into smaller molecules by our digestive system. Carbohydrates are broken down into glucose, proteins into amino acids, and fats into fatty acids. These smaller molecules will then enter different pathways or different reactions in order to produce ATP. In fact, the production of ATP is done through two methods. The first method is called substrate-level phosphorylation. This process does not require oxygen, but it can only produce about 4 ATPs per glucose molecule. Substrate-level phosphorylation is a direct ATP generation that involves the transfer of a phosphate group from a high-energy substrate to ADP to form ATP. Some examples of substrate-level phosphorylation can be found in glycolysis, specifically the conversion of 1,3-bisphosphoglycerate into 3-phosphoglycerate and the conversion of phosphoenolpyruvate into pyruvate. Another example of substrate-level phosphorylation can also be found in the conversion of succinyl-CoA into succinate, a reaction that occurs in the citric acid cycle. The second method is called oxidative phosphorylation. This is the primary source of ATP production. It does require oxygen, but it can produce up to 34 ATPs per glucose molecule. Oxidative phosphorylation occurs in the mitochondria of a cell. So inside the process of oxidative phosphorylation, electron carriers like NADH and FADH2, which are products of the citric acid cycle, will donate electrons into the electron transport chain, which is a protein chain located inside the inner mitochondrial membrane. And as these electrons travel down the protein chain, moving from one molecule to another, it pumps proton ions into the intermembrane space, creating what we call a proton gradient. This proton gradient will cause the proton ions to move back into the matrix, and an enzyme named ATP synthase will use this movement to generate ATP. This process requires oxygen because at the end of the chain, oxygen is needed to grab the electrons and combine it with proton ions in order to form water. So now that the production of ATP is completed, let's proceed to how it is used. Most people refer to ATP as the energy, but to put it more accurately, ATP is not the energy itself. It is more of an energy-carrying molecule. ATP is a highly unstable molecule because it consists of 3 negatively charged phosphate groups which repulse against each other to produce a high-energy bond called the phosphonitrate bond. So when an ATP goes through hydrolysis, it will lose the phosphate group furthest away from its ribose sugar. And since this is a favorable reaction because of the repulsion between the negative phosphate group, this process releases energy. One book named The Essentials of Human Anatomy and Physiology by Alain Marier actually described this perfectly. The book quoted, "The high-energy bonds of ATP are like coiled springs that release energy for use by the cell when they are broken." So after the energy is released, how is it actually used? The phosphate group that is released during the hydrolysis of ATP will then bind to another molecule, making it highly reactive and unstable. Most cells will use this feature as an advantage by linking two types of reaction together to form what we call coupled reactions. It links the exergonic reaction, which releases energy, with the endergonic reaction, which uses energy. And since ATP hydrolysis releases energy, it is considered an exergonic reaction, and it is often followed by the endergonic reaction, which uses energy. Let's take glutamine synthesis as an example. The exergonic reaction is when the phosphate group is separated from ATP and binds to glutamate, forming the highly unstable and high-energy intermediate called gamma-glutamyl phosphate and ADP. The anorganic reaction is when gamma-glutamyl phosphate reacts with ammonia, displacing the phosphate group from its molecule to form glutamine. Glutamine synthesis is just an example of how energy can be used to fuel chemical reactions. In fact, energy can be used to serve many other purposes, such as transportation, where energy is used to transport solvents, such as amino acids, across cell membrane. Muscle contraction, when it is used to activate contractile proteins which allow muscle to contract and relax. And the synthesis of macromolecules, where it is used to synthesize larger molecules such as DNA or RNA. What makes ATP special is its ability to be continuously recycled, ensuring steady energy supply to the cell. ATP provides energy by breaking down those high-energy phosphonitrate bonds between negative phosphate group, leading to the production of ADP and a free phosphate group. But when energy is needed, ATP can also be resynthesized using the ADP and the free phosphate group produced during the ATP hydrolysis. This cycle is known as the ATP-ADP cycle. And since ATP hydrolysis releases energy, the resynthesis of ATP is the opposite. It requires energy. And this energy can be derived from glycolysis, it can be derived from beta oxidation, or any breakdown of larger molecules that can aid in the reattachment of the free inorganic phosphate group to ADP in order to form ATP. This continuous cycle ensures sufficient energy supply to support necessary processes of life. So, in summary, ATP is an energy-carrying molecule that consists of a nitrogenous base, a ribose sugar, and three phosphate groups. It can be produced by two methods: substrate-level phosphorylation, a process that does not require oxygen but can only generate about 4 ATP per glucose molecule, and oxidative phosphorylation, a process that is considered the primary source of ATP production. It requires oxygen but can generate up to 34 ATP per glucose molecule. ATP releases energy after high-energy phosphonitrate bonds between negatively charged phosphate tails are broken down, releasing ADP and a free inorganic phosphate group in the process. This energy can be used for fueling chemical reactions, transportation, muscle contractions, and the synthesis of macromolecules. ATP can also be recycled from ADP and a free inorganic phosphate group through a process known as the ATP-ADP cycle.